Hello everyone. Welcome to week 8. We are reaching there. Okay, as soon as I hit the recording button, again, something has wrong with the Zoom. Stay with me, guys. Hi again, everyone. Sorry for that little flick on my Zoom. Welcome to week 8. And today we are talking about nutrient control of gene expression. So I hope you have revised your previous knowledge about DNA, what is gene, what is it made up of. And I noticed just a few minutes ago that some of you have gone and done this post-lecture activity, which is really great. Well done. So my aim is that we'll go through DNA structure. We'll talk about purine and pyrimidine. Quite basic. We are not delving deep into each reaction in these pathways, but I'll highlight some of the enzymes and some of the cofactors as that is relevant to some of the genetic or hereditary disease condition. Then we'll talk about telomeres. I know I've missed writing it here. Then come to 1-carbon metabolism. That is now, you all know that it is transfer of that methyl group from folate and how it is used for epigenetics and other mechanisms in the body. So we'll touch upon what are the various epigenetic mechanisms that human use. However, we'll stay with methylation. Okay. So we all know that people who discovered the structure of DNA, Watson and Crick, long back in 1960s, they received Nobel Prize. So and the DNA, that is deoxyribonucleic acid, they are the bases. And each nucleotide is made up of deoxyribose and a phosphate group and a nitrogenous base. And these nitrogenous bases can be purine or pyrimidine. So purines are adenine and guanine, and pyrimidines are cytosine and thymine. Okay. Just a little more picture to show how that DNA is kind of combined as a double helix structure. So we have a sequence of A, G, C, T going on so that we have the entire DNA structure. Would you like to comment then that what is a gene then? What is a gene in a DNA?
It's a part of the DNA?
Yes, correct. Yeah. Thank you. Anybody else?
It's a small part that determines like the shape, the size, of a certain part of the body.
Okay, yes, you are almost there. So it is a group of these nucleotides that are together and they translate for a molecular structure or a function in our body. So on our DNA, we would have particular set of genes that are transcribing to make insulin, or they are responsible for making folate enzymes. Okay. Yes, Isabel, you're right. It codes for a protein. Yes, that's correct too. So how is our body doing this? So we have a de novo process, that is when we can synthesize purine and pyrimidine from the basic molecules such as carbon, nitrogen. That's what we call as de novo mechanisms. However, as I always mention, our body knows how to conserve. We are really sustainable. So there is a way called a salvage pathway in which from the end product, that is AMP, GTP, so we make GTP from guanine, ATP from adenine. So from these end product of this entire process, if the body notices that I have excess of it, then body salvages it. Body utilizes it to make the original structure. Yes, de novo means from scratch, from very beginnings. So this slide is actually a very little summary for the entire purine synthesis by de novo pathway. Why it is a very small summary? Because this actually, the whole process comprises of about 12 to 13 steps. And I don't want to scare you away to having to remember all that. Plus, that is not going to be relevant in the near clinic practice, unless you want to really go into an intensive gene testing mode. Now, the point to remember from this slide or this pathway is, where does it all start? So it starts from PRPP, 5-phosphoribosyl 1-pyrophosphate. So this is the first compound that the pathway uses, and from there it adds a folate group, it adds glycine, it adds aspartate, it adds glutamine, and then it goes on to build its structure. So IMP is the intermediate compound, inosine monophosphate. From there, body makes adenosine triphosphate and guanosine triphosphate. Then it also goes on to incorporate this into DNA structure. So from this one, just remember this first starting point for purine de novo synthesis. Now this slide, I want to mention 2 points here. One is that when I am starting with PRPP in everybody, whenever we are making this DNA, there is a negative feedback loop happening even here. So when body knows that there is too much of AMP and GMP that are being generated, they can go back and inhibit their own production. Why do you think body is doing this? Trying to think, what does body do with DNA?
Make a copy.
Sorry?
Make a copy.
Yeah, make a copy. And what will happen if we have too many copies, too much DNA?
Nancy, is it like a cancerous growth?
Yes, thanks, Kathleen. Welcome.
Thank you. Hi, Nancy.
Yes, that's what I wanted the discussion kind of little to go on. So body knows that too much of DNA can be a problem. It can go towards tumor. And again, I'm really simplifying it here. So there is a negative feedback happening even if when we are making and metabolizing this DNA. So AMP, GMP, which are the end product of this pathway, not end, intermediary products, they can inhibit PRPP, which was the first compound from which all this de novo synthesis started. Okay. Then there's one more point. We are not going into detail about the salvage pathway. However, just a small mention that salvage also means that body again does not want excess of these accumulating anywhere. So body can go back and make purine nucleotide from its end products again, so that we don't have excess and we don't pressurize the kidneys to excrete all this DNA remnant. That's why this salvage mechanism is in place. And there is a different enzyme. It will come up later in the slide. Let me just go back here. This is the enzyme. So hypoxanthine phosphoribosyl transferase. If that enzyme, which is controlling to make purines from its end product, there are genetic mutations in this pathway, then people can have various disease conditions such as gout. And I'll again come back to gout in a second. So as you can see, this slide is talking about how we are making DNA or RNA from a scratch. We are using nitrogen and carbon donors. So from glycine, my cells can take nitrogen and carbon. From 10-formyl tetrahydrofolate, one of the various amazing form of folate, I use carbon group CH3. Similarly, from carbon dioxide also, we can take carbon so that we can continue to make purines. And these are some of the other cofactors, and I encourage you to remember some of these cofactors. So now what happens if there is an issue with purine metabolism? So when we have made adenosine into AMP, guanosine into GTP, then the process goes on, body tries to use it. If body cannot use it, then the end product is uric acid. So if we are making a lot of purines inherently in our body, if for some reason there is enhanced catabolism of nucleic acid, or I'm just through excessive intake taking a lot of purines, that may lead to hyperuricemia. And one of the conditions that we are aware of is gout. So it can not only happen when we have mutation in the salvage enzyme, it can also happen if we are consuming a lot of purines. Any questions at this stage? Am I going too fast, or am I going slow, or is it easy? You already knew a couple of these things.
I think you're going well. I think it's good.
Okay. Thank you. Thank you. Right. So tell me, what do you know about gout? You can click on this link and try to read. If anybody has already read about it, has dealt with gout, what do you know about the foods that we need to omit?
Nancy? Oh, sorry.
Sorry.
You go, please.
Oh, no, it's fine. You go.
Well, I was just going to say, I know alcohol is a big thing, Nancy, with gout. And is that just through the pathway that the purines are? Is it the uric acid pathway, Nancy? I'm not sure, but I know that alcohol, they say that generally that can be a...
Yes. Yes, you're right. That's the pathway as well. So alcohol is people are asked to let go of alcohol if they are susceptible to gout.
I think acid foods as well.
Yes.
Yeah.
Yes, that's correct too.
Seafood, possibly?
Sorry? Yes, seafood. Yes, correct. Absolutely. Yes. And does anybody know which ones among seafood as well?
Prawns, wasn't it?
Yes. Yes, it is the prawns. Yes. And I'm not sure if you have read something known as Syndrome X. I might have mentioned it earlier. Metabolic syndrome or Syndrome X. They say that people who are insulin resistant, they are also susceptible to gout. And we still don't know why and how, but it is a major player. So just being mindful that if you have a client coming to you with gout, flare up, then we need to control their insulin as well.
And Nancy, is it silly to ask, but prawns, why particularly prawns? Are they just high in purines or?
Yes, they are high in purines. Yes,
Purines.
that's the point.
Okay.
Yeah.
Okay.
So like
Thanks.
if you compare, they all are high biological value protein, like chicken and prawns,
Yep.
but the content of purine is higher in seafood and especially in prawns.
Okay. Thanks, Nancy.
That's okay. So moving on with pyrimidine, that amide group from glutamine is condensed with carbon dioxide to form this particular compound, carbamoyl phosphate. So that's the first compound. Like PRPP for purine, for pyrimidine we make carbamoyl phosphate. And then we are making orotic acid here. And the reaction is catalyzed by carbamoyl phosphate synthase 2. And you don't have to remember this one because it's a bit of a tongue twister. But then we are making uracil from, first we make cytosine and then we make, sorry, I'm saying the reverse. Uracil is the one we are first making, and then we are adding glutamine to make cytosine. And in the process, we are using serine, glycine, lysine, and of course, folate in this synthesis. There are a few inherited disorders that are a result of pyrimidine biosynthesis. And we call it as orotic aciduria, in which there is a megaloblastic bone marrow, erythrocytes are hypochromic, and it is also characterized by leukopenia. So there may be retarded growth in the muscles, and eventually people may even have hepatic encephalopathy. We don't know if much can be done to prevent this condition. Okay, so that was just kind of an overview of purine and pyrimidine synthesis and some of the enzymes and cofactors that we use to do so efficiently. Coming on to telomeres. What are these telomeres? Anybody knows? So, you know, in our DNA, we have got genes that are coding. That means they are coding for a particular protein, as you mentioned earlier. Telomeres are actually nonsense codon. Okay? They don't code for anything. And yes, Isabel, you are right. They are kind of tails of our chromosomes. But they don't code for anything. So what are they there for? Okay, let's know more about telomere. So like in my shoestring, we have got, in the end, we have got a solid structure which protects the entire structure not to get unraveled, not to get messy. Otherwise, all those threads keep coming out of my shoelace. So it's something like that. Okay, and this analogy has stayed with me since I heard a professor speaking in a conference. So it's kind of an end of telomere. They have a repetitive sequence such as AATTCC. It is quite similar, but it does not code for anything. But it is essentially there to protect chromosome from degradation activities. However, every time a DNA replicate, telomeres are shortened. See, it's a quite complicated process when DNA are replicating. So sometimes some of the genes can get lost. They may get degraded. However, instead of genes, we have got this excess material that gets degenerated in the process. So these are the sequence, TTA, GGG, and it is actually repeated 100 to 1,000 times in different chromosomes. So why are we studying about them? Why is it important to that we know that we have something called as telomeres? There is data that when we are changing our environment, when we are putting pressures on our DNA replicative pathway, these telomeres are getting shortened. If they are, and that process is actually happening quite quickly. And we are now associating telomere shortening with various chronic diseases. So if there is stress, that's why I said listen to the music. Because when we are going through a chronic stress, then telomeres get shortened. Okay. And there is this thing written, telomeric fusion. So what happens is that if there is not enough telomere, when the chromosome is replicating, instead of getting separated properly, they get fused. And that's when we get aneuploidy or mutations happen. And if that accumulates, that becomes a cancer risk. However, we now know that if we are containing a lot of eating diet rich in antioxidants, if we are doing exercise, yoga, meditation, then somehow they are able to preserve the telomere length. So all this research about telomere has been associated a lot with preventing aging. In fact, one of the researcher group in Boston found out that babies who are breastfed for a longer time, and then when their telomere length was measured when they were 6 or 7 years of age. So breastfed babies had a longer telomere length as compared to babies who were not. So whole lot of lifestyle factors, dietary factors are being studied to understand telomeres. So it's now proposed that it represents that molecular clock, and that underlies aging. So there is a whole lot of research to understand how we can prevent it and how we can keep them long so that aging and aging related disorders can be prevented. There is sufficient data that telomerase shortening, telomerase is the enzyme that's responsible for telomere length. So this should be telomere shortening and not telomerase. So my bad. So telomere shortening induces chromosomal instability. And I gave you an example how that happens. And that is now associated with cancer, stress, dementia, CVD, and obesity. And then a lot of data coming from Adelaide CSIRO Nutrigenomic Lab, because I was there and I saw it all happening. So they did a lot of experiments in the Petri dish, and they allowed telomeres to grow in the presence of different concentration of folate and even B12. So they looked at various nutrients, and what has stood out and what has faced the research, and we know that low folate level is associated with shorter telomeres. And we'll know why, because folate is not only used for formation of DNA, it is also used for epigenetic modifications. So that's kind of an interesting fact that has stood the test of research. Any comments, any question at this stage? And yeah.
I was just going to ask, like, it's got the folate, but if the methylation pathways were underperforming as well, would that possibly shorten telomeres as well?
Yes, Jacinta. Yes. And we will learn about that soon.
Okay, cool.
Okay?
Thank you.
Good.
Nancy, I was just going to say too, when you chatted about low folate, you think about, in pregnancy, the neural tube defect, isn't it?
Yes.
In folate. So you think, oh yeah, that obviously it all links up, doesn't it?
Exactly. Yes, Kathleen, you're right. And we are still not testing telomeres, okay? But I have kept this, just 3 or 4 slides in this session so that when we become practitioner, we are able to educate our clients about benefits of healthy life and lifestyle and dietary habits, that it can modulate at even DNA level.
Nancy, how would they test for telomere length anyway?
Yeah, so we are not doing it generally for blood testing, but in the laboratories, we are able to separate telomeres from our WBCs. So we take blood, do centrifugation at a particular speed, then we separate lymphocytes from the red cells, and then those lymphocytes are passed through PCR machines, okay, where we multiply DNA And from there, we are able to look at telomerase concentration as well as the length of the DNA. So we see it under big machines.
And
Yes. Yep.
say you said you're looking at lymphocytes, but
Yes.
would that say if there was shorter telomeres on that strand of DNA, would that mean that it would be throughout with all cells in that body, or?
That's a good question. We can't generalize like that. Yes, but kind of that gives an indication that lymphocytes are responsible for immune function, for healthy body. So if they are compromised, they're a good chance of extrapolation that it's happening with other cells. But there is a chance that there can be discrepancy there also.
And I guess they're dividing rapidly, aren't they? The white and red blood cells in the bone marrow. So we're producing more of them. So
Yes.
No, thanks, Nancy.
Good point. Okay, now Isabel is asking something. How to draw the middle line? Yes, that's tricky. What do you all say about that? Balance isn't, I hope like our journey in this biochemistry is making us appreciate that the whole effort of our body is to have that balance. And it is done at a micro molecular level. So maybe just listening to our body, just not running after supplements all the time unless we need it, correcting the environment so that our normal body processes can thrive. But I don't have the exact answer, Isabel. But we do try to listen to our body, isn't it? Okay. Do we need a break at this time? How are we going with time? Because my screen is kind of shifted today. I can't see many things. Right. We are cruising along fine today. So today, now we are going to talk about 1-carbon metabolism. It is mediated by folate cofactor and it comprises a series of interlinking metabolic pathways that include methionine and folate cycle and then sulfuration pathway. This is happening in every cell of my body, okay? And why my body needs this is that I need to move around that carbon molecule from one compound to the other so that I can synthesize purine. I can have a homeostasis for amino acids, and I can use that transfer of methyl group for epigenetic maintenance and keep a balance of redox, that is a balance between antioxidants and free radicals as well. So there are major functions of this 1-carbon metabolism cycle. So please remember these functions. Now I've added few animations here. And if you don't like this diagram, you can search on articles on PubMed, on databases. There are different version of the same pathway. However, I made this when I was doing my PhD, when I was learning about nutrigenomics. That's why I have stayed with this colorful one. I'll go really slow here. You don't have to remember this pathway. You don't have to remember the name of the enzymes as well. But when we are doing epigenetic, I'll share one of my genetic gene testing report for my own self, and there you will understand why each step is important in this pathway. So remember, when we absorb folate, we convert it into a monoglutamate form, and then it comes in the blood and it reaches liver, from where it is transported to every cell of the body, because every cell needs it. So when that, I know I have written folic acid here, just to indicate that it is the monoglutamate form that is starting this reaction. So that monoglutamate form is converted to dihydrofolate. How? With the help of this enzyme, 5,10-methylene tetrahydrofolate dehydrogenase. Okay. Let me remove that one. And here, where is that extra carbon coming from? It is coming from serine. Okay. Serine itself gets converted to glycine. And in the presence of B6, I make tetrahydrofolate. You might have seen that now we have another form of folate being sold as a supplement, which is called as folinic acid. So this is the one that they talk about, 5,10-methyl tetrahydrofolate. Why it is important? Because from there I make the active form of folate, that is, 5-methyl tetrahydrofolate. Okay, so that's why they say folinic acid is better. Now I want you to look at some of these enzymes. So one I already mentioned, MTHFD1 dehydrogenase, and the other one is MTHFR, which is the most common researched and most talked about enzyme. Why it is important? Because using this enzyme only, I can make the active form of folate, which is 5-methyl tetrahydrofolate. And to do so, I need riboflavin. That's why, it's important that we supplement with most of these active Bs together, because they need each other at this level. Please stay here only. MTHFR is important because from this pathway only I am making thymine DNA that goes on to participate in making my DNA. And here also I make my DNA repair enzymes. So if somebody has a mutation, has a SNP, and I'll talk about what's the difference between two in just a second. So if there is an issue here, then those people may not be having a good functioning of this enzyme. Its capabilities may be reduced by 50%. It won't be efficient. So there would be interference with all the pathways, because this is happening in every cell, in the neurotransmitter, in my liver cells. They are all going through this pathway. Now we know where is MTHFR and how does it play important role in different disease conditions. Okay, so now let's move on. This is the folate part of this pathway. Okay, now it merges with methionine pathway. How? Because this active form of folate, in the presence of B12 and another important enzyme, MTRR, it makes methionine. And we know how important methionine is because we make proteins, and methionine is one of the important amino acids. Now, in regard to autism spectrum disorder, there is a lot of research telling us that there may be mutation in this pathway, MTRR, methionine synthase pathway. The names are written in the next slide. So that's how importance of OCM in autism-like condition as well. So they are saying maybe we need to supplement with B12, and that is able to modulate some of the symptoms with autism spectrum disorder. Moving on, when we have reached methionine, again a very, very crucial step, because body has to use methionine to make SAMe, S-adenosyl methionine. Why? Because SAMe goes on to participate in methylation reactions. That is when we are expressing different genes. So if somebody has SNP in either of these enzymes, either MTHFR, MTHFD, or MTRR. So it's not only just one enzyme, it can be mutation in different, and this pathway can be disturbed. But how do we know that this pathway is disturbed? One of the main parameter that we can measure to know that this pathway is not working properly is when people have high homocysteine in their blood. So homocysteine is actually an intermediary protein. When we are making methionine to SAMe, we make it as an intermediary compound, but it does not have to stay there long. Either it can be converted back to methionine in the presence of MTR, or it can be converted to cysteine in the presence of B6. Now, do you remember what we make from cysteine?
I think we make glutathione. Oh, yes.
Yes, and what is
There is-
glutathione? I keep going on and on, but we make our own antioxidant from the same pathway. So if I'm doing everything right, but I'm deficient in B6, both these pathways would be inhibited. Maybe I don't have MTHFR mutation, but if I have MTR, then also these pathways would be impeded or inhibited. Okay, I'm going to shut up now and take questions. I hope I have not confused you, but let's stay here for a while and think about how many ways that this pathway is affecting our different body mechanisms.
Nancy, can I ask you, which enzyme was it that they associate with autism? Was it the
MTRR.
MTRR? Yeah, thank
So
you.
they are saying there's some, when they supplement with B12, the symptoms subsided. So, and then they found out that it's not only MTHFR, those children may have MTRR mutation. So it is preventing, if there is this mutation, then B12 cannot participate further in the reaction. You can see there are double arrows most of the time.
Yeah.
So either pathway would be
Impaired. Mm. So Nancy, this is going on in every cell, this one carbon metabolism?
Yes. The day I realize it, carry on.
In the nucleus or?
It's happening in cytosol as well as mitochondria.
Mitochondria and cytosol. Okay. See, thank you. And Nancy, when I look at all this, I think it's a wonder we're all functioning, isn't it?
It is yes.
It's a miracle. Mm. Thank you.
Okay, Samantha is asking something. So is it only thymine that is produced in this pathway? Yes. Yes, Sam. So other bases we are making from other sources, pentose pathway and others. We are not learning about that. And in fact, I don't remember some of them, so sorry. Now you asked that, I'll just share one more thing. Can you see that I am making TTP from UMP here? So from uranidine monophosphate, I'm making thiamine triphosphate. Sometime when somebody is having a lot of DNA damage because of, so many factors, then instead of thiamine, uridine get incorporated in the DNA. Okay. So because, body is trying to function, it is trying to make use of something. However, that changes the gene sequence, and that in turn impact the functioning of whichever protein that gene is making. So that's another thing which they can measure, because, all these metabolites of uranidine and thiamine, they all reach urine. So they can measure DNA damage in the urine by measuring the catabolites of these pathways. Any other question? Do you want to?
When you said that one carbon metabolism is in every cell, and you said it was the mitochondria and something else?
Cytosol?
Cytosol. Okay, thank you. I had another question. When it says methionine goes to protein synthesis, is this like, because I'm into sports nutrition, this isn't just protein synthesis for muscle building, this is to create new cells, is that right?
Yes, correct. Yes.
Thank you.
Great. No, all good.
Nancy, can I just ask you, I'm a little bit confused. Just when, when you said, oh no, this is the way that this is, so we only make one of the bases through this one carbon metabolism. Is that right?
Yes, yes.
So when it says that its functions up the top there, Nancy, of one carbon metabolism, it says biosynthesis, it says purines and thymidine.
Yes.
And adenine and guanine, but it doesn't mean that we're making them. Is that right? We're just making the thiamine. I'm just a bit confused, Nancy. I was all right until I realised.
No, Isabel is also asking the same thing. See, via this pathway, we are making the DNA requirement of thiamine. So that's where, let's go back a little, guys. And I'll go to the first, here. Can you see tetrahydrofolate? That's where in the structure, I am making one of the purines, and then from the same, I'm also making adenine. So it's a cofactor there. But in most of these slides, we are talking about DTTP because that's where we notice mutation happening. And that's maybe what we are looking in evidence. We have not looked at the other purines because we can make that from here.
But
it goes on and build on this pathway.
So Nancy, when you say the other purines, but thiamine is a
pyrimidine.
Pyrimidine.
Yes. Yes.
So yeah, I'm still.
We are studying only this one. We are not going towards this one. Can you see here? We are making, so from the same pathway, we can use that tetrahydrofolate to make purines as well.
Okay. So yeah, I'm sorry because it just says.
No, that's fine. That's fine. Even it's
I'm confused. I think because thiamine's a
pyrimidines.
But then we're chatting about purines. I think I'm just totally confused.
Yes. Because from the first part of this pathway, this tetrahydrofolate is the one that works as a cofactor for making purines.
Right.
And then kind of the similar one we can make for making pyrimidine. So this is just one example.
Yeah.
That's why an arrow that we are doing more DNA synthesis from this branch. So just trying to emphasizing various branches the same pathway can take.
Okay, so more or less it's just showing, Nancy, that sort of how interlinked these pathways are. Is that right?
Yes, and how pivotal it is, you know.
Okay.
And I've just added this one here. Again, don't want to confuse you, but a couple of years ago, I had a student and he had a mutation in the CBS gene. Okay. And that's why I've just added this one, that when you go in a clinic and if you are reading a gene testing report, you may have a client who has an issue with CBS. That's telling us that from that same pathway, we are making glutathione. So if there is a mutation in CBS, then this branch of the cycle is impacted. So we are not able to make cysteine, we are not able to make glutathione, and people may be susceptible to free radicals and other diseases. Okay.
Because Nancy, I have a heterozygous, I think I've got a gene defect in MTHFR.
Mm-hmm.
Yeah, I mean, I found that out a long time ago, but you just think, oh, I said, I know it's only one, and I think that is quite common from what I've heard, but I don't know, Nancy. Just see this pathway. So my pathway would be a little bit impeded there.
Yeah, see, this area is again under more research.
Yeah.
What they are trying to do is they are combining the data, among Italian population, among Caucasians, among Asians. So they are trying to look at from that perspective as well. And again, I'm talking just one of these. There are several mutations. There is one that is called as C to T, the main one, but there is another one where instead of adenine, there can be a guanine in MTHFD1 gene. So it's different because everybody is unique, everybody is exposed to different condition, they have a different genetic history. So we are actually now finding out there is not just one polymorphism, there can be many more. Okay. And when you said that it is quite common, the current data is that only 10% population has it. Okay. So I'm just skipping and coming to the SNP. Oh, sorry. So we are talking about polymorphism. So I thought I'll just add this slide that SNPs or single nucleotide polymorphism, they are common type of genetic variation. And remember when I asked you about what is a gene, okay? And you said that it is a small portion of that DNA which is making a particular protein. So when there is a change in the nucleotide chain in that gene, that changes the structure of the protein. That is what we called as SNP or single nucleotide polymorphism. But tell me, does that mean that SNP is a mutation? What do you think?
Nancy,
Yeah.
I don't know, maybe my opinion would, because it seems like if the gene just always codes for that,
Yeah.
I think a mutation was something that perhaps happened, not frequently, or it was a one-off sort of a thing, and then that just goes on to mutate and mutate and mutate. But
Yes.
I perhaps if there was a change in the DNA coding, I didn't know if I would call that a mutation, but I could be incorrect.
No, 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 you are kind of on the right path, and that's why I have added this slide so that if I'm not able to communicate correctly, something is written and we can make inference from there. So see, SNP is always happening in different genes because it is such a complicated machinery, isn't it? And so something or the other keep happening, but our body is very intelligent. If there is just one change in the nucleotide, A, G, or T, C, body can still make the same protein. But you can understand the efficiency of that protein would not be the same, isn't it? Because there has been a change in the structure. It may be a minute thing, okay? But if there is a mutation, that is actually when the end product completely changes and it fails that particular person because that pathway is completely not working properly. And I'll give you a very, very simple example. And you can laugh, but kind of, I don't know why I think about such examples. So let's say that I have learned to make risotto, okay? And I am told that it uses a particular type of rice. I have to add particular amount of cheese and wine, okay? So that's the basic solid recipe that has been used for ages. But when it come to me, I change it slightly. I use a different kind of cheese. I don't add wine in it. But I'm still making a risotto, which is less tasty and less efficient. However, if I change the rice completely, then I'm not making risotto, I'm making something else. So in a way, when I'm changing it drastically, that the end product is not happening, that's what a mutation is. Have I made sense, or I've really simplified it and messed with the example?
I personally really needed that. Thank you.
Yes, good, Nancy. Well done.
Okay, great. So yes, Jane, you are right. SNP can be supported by epigenetic modulations, and mutation kind of is definitive. And we still don't know. I know we are doing a lot of advancement in stem cell therapy and all that, but we are still long way from correcting mutation. Absolutely right, you are. Okay. Now, why we are reading about one carbon metabolism again? Because SAMe, I think I've talked a lot about SAMe earlier also. So polymorphism in any of the enzymes in this pathway that we studied, and deficiency of any of these vitamin Bs would end up having a low or suboptimal functioning of OCM. That may lead to elevated homocysteine, which is now known as an independent risk factor for inflammatory conditions such as hypertension, CVD, preeclampsia. But still, nobody measures it. A GP won't measure it. I don't know why, whereas it is such a clear indicator. Now, you might be thinking, can we correct it? Yes, if we notice that there is high homocysteine, and I'll again go back to the diagram. If I supplement with B6, if I supplement with B12 and even B2, there is research evidence that I can reduce homocysteine in the blood and I can reduce inflammation. Okay. So if you're asked in the exam, list function of 1-carbon metabolism and what are the implications for its poor functioning, then we are asking you to comment on different disease conditions that can arise because of polymorphism in the enzymes and cofactors in this same pathway. And this is what we know so far. Okay, Deborah is saying they don't measure it because they don't have a clinical pathway to respond to a high homocysteine reading. They know it, clinicians know. There has been a book written by one of very experienced GPs on homocysteine. And
That was what my GP told me, Nancy, because she ended up writing me a request for a homocysteine reading, a blood test, and she said, well, maybe your naturopath can do something with the results, but as a GP, she didn't have a response, because I guess they don't prescribe vitamins.
Yes. Because they want to fix it with a pill.
Correct. Yeah. It doesn't have a pharmaceutical response.
Yes, Deborah. Yeah. And every time in the student clinic, students come to me and ask me, why can't we get homocysteine measured? And I'm like, I really don't know. Okay, maybe hopefully soon it will come up. Okay. So when I was talking about, the polymorphism, and Kathleen asked me how common it is. So the common one, MTHFR, that we know is when instead of cytosine, I am adding thymine in the blood. So, and that changes the formation of this enzyme. Okay. And this is what we know as that there is a substitution at the number 677 nucleotide. So that's how long the gene is, How many nucleotides? 700, 800. And by experiments, we know that it is the number 677 which is an issue. Okay. And these people would have high homocysteine. And the other one I have not added in the slide, it is called as AG. Okay. Now, if you will learn about these DNA tests, then they do mention it that it is MTHFR1 and MTHFR2. And they are able to measure both now in gene testing. So elevated homocysteine is independently associated with all these conditions. So that's why I'm not happy when they are delaying introducing it as a normal test. Isabel, I think they are doing it not via Medicare, they are doing it by other testing. So there they can measure it, but we have to pay for it. That's the point.
But Nancy, it seems silly, doesn't it? Because if you find out you've got high homocysteine, supplement B6 and B12 could help that.
Yes,
much better for people suffering from chronic disease. And at the end of the day, it's a much more economical way of helping people, isn't it, as well? Not only better for their health, but seems crazy, Nancy.
Yeah. Sometimes you feel that it is so simple. There is a simple solution. Why do we go after complicated things? Okay. We don't have answers so many times. So through research, now we know that these are the nutrients that can reduce homocysteine because they will ensure that pathway is functioning in a normal manner. So please remember some of these nutrients as well from exam perspective. In the short answer question, you can be asked, list 3 nutrients that can reduce homocysteine. Okay? So please remember that. Okay. Let's take 10 minutes break, guys. Is it okay?
Yes, ma'am. Thank you.
Yeah, so let's restart. We are now going to talk about epigenetics, and we'll now know how that methylation pathway, how that 1-carbon metabolism controls whether a gene is expressed or not. So all the cells in our body, they have the same DNA. Yet a stomach cell is digesting food while a heart cell is pumping blood. Similarly, genetically identical individuals, they have the same DNA, yet they develop into different people with different preferences and different behavioral traits. So clearly, there is something other than genetic makeup. And that's what now we understand as epigenetics, which literally means above genetics. And it's a fairly recent knowledge. We came to know about it in 1997 after famous experiment by a few researchers in US, and I'll talk about their study in coming up slides. So these epigenetic factors, they are working throughout the lifetime, and we now know that they are passed to future generations as well. So what are these epigenetic mechanisms? They are independent from structural DNA sequence, and they control gene expression via chromatin remodeling system. That is when the chromosome is opening up. Then there are post-translational histone modification. There are different types of proteins that unravel when a gene is to be expressed. Then there are certain known coding RNA function, but we are focusing our attention only on DNA methylation. That means when SAMe is made from that folate methionine pathway, it has got a methyl group to give away. So that methyl group, when is added or removed at the end of a gene, that decides whether that gene is expressed or not. Okay. So all the cofactors in 1-carbon metabolism become necessary or become cofactor for epigenetic mechanisms as well. So what this diagram is kind of detailing is each aspect of these epigenetic mechanisms. And what it's also showing is that they are impacted by different environmental factors, even in utero. And I did mention about early origins of metabolic diseases. There also epigenetics is playing an important role, that when a baby is growing up or whether when a lady is preparing for pregnancy, then environmental factors can switch on or off a particular gene. So In DNA methylation, again, I'm repeating myself, methyl group can tag DNA and activate or repress it. Does anybody has a query at this point? I'm asking because earlier there was a notion that methyl group always has to attach to a DNA for that gene to be expressed. However, it is a little more complicated than that. We still don't know all the genes that are being expressed by addition of methyl group, and some are expressed when that methyl group is removed. So we are still learning about that. And remember, I shared my example of working with the placental tissue. So in that small review, I found that in the placental tissue of a pre-eclamptic women, there were different types of methylation. Some of the inflammatory genes were expressed when methyl group is attached, and some were expressed when methyl group was removed. Okay. So the point that I'm trying to make it that either way, the genes may or may not be expressed, and we are still learning. We are still not sure about which gene is expressed by adding a methyl group and which is expressed by removing a methyl group. But we know that various factors and processes that can impact people's health, exposure to metals, environmental toxin, various drugs, physical activity, that all can prevent these diseases because we can modulate this expression of gene by those change in habits and dietary interventions. How we'll know soon. So at every stage of DNA synthesis, one carbon metabolism, and epigenetic pathway, all these nutrients are playing major role. And some of the cofactors are listed here for you to remember. And what happens if DNA's health is damaged? If there is an issue with gene expression, how do we know? There are various ways of testing DNA damage, and one of that is measuring micronuclei. And remember, I told you I'll show you my own lymphocyte. So this is my own lymphocyte. And what you can see is that mitosis has taken place. So the cell has divided. And when it was dividing, can you see these small 2 dots? So this is the leftover DNA that failed to get incorporated when the DNA was dividing. And this is called as micronuclei, and there is a lot of data that more is the number of micronuclei, more is the DNA damage, and that is observed to be found in women with preeclampsia, with certain cancers, even mental health, infertility, those kind of placental insufficiency. So in all those kind of condition, if people have more micronuclei, that indicates that there is DNA damage, and that leads to origin of these diseases. So can we prevent it? And how do we prevent them? So this is that famous experiment which I was talking about. It was done in 1990s, published in 2003 by Waterland and Jertel, where they again sacrificed few rats. So what did they do? So they were trying to understand whether mother's diet can change the epigenome of the offspring. So all the rats, they have got this particular gene called as agouti. I really don't know how to pronounce this correctly, but I call it agouti. Now this gene is unmethylated, okay? And when somebody has this gene, its coat is yellow, and rat is obese and is prone to diabetes and cancer. Now, when agouti gene is methylated, it is normal. The rat is brown and it does not develop diabetes or it is not obese. So that was kind of the basic start for this experiment. So what the researcher did was that they fed this mother who was yellow and who was big, they fed it with folate and choline and B12. And what they noted was that the babies that were born were thin. So by dietary modulation, they were able to switch on that agouti gene and change the disease, prevented the disease to occur in the progeny. Questions? What do we learn from here, or what do we, yeah, take it from here?
That it can be reversed, like it's not a.
Yes, Victoria. Yes. So we can, as somebody said earlier, that some things are in our hands. Yes, Isabel, dietary modulation is powerful. Yes. Yes, Jane, there are human studies as well. Because as you can see, this was done about 20, 30 years ago. Okay. I'll come to the human studies in a second, but does anybody has any other question? So we understand how epigenetic is different from genetics and at what level we are interfering and modulating. Now you understand even that methylation. We can work around histone and other things, but I'm not talking about that because it has still not translated into clinical practice. Okay. Yes, Isabel, I completely agree. What all this understanding just tell me is that yes, our connection with environment and food and herbs, I think we are just witnessing what all we already knew. Okay, now Victoria is asking, dietary modulation can reverse gene expression. Yes, Victoria. And so I'm going to show you something. Okay. And I must thank FitGenes. And that's because that's courtesy them. I have my report. Okay. I'm also not saying that this is foolproof. This is just an indication to understand how dietary modulation can switch off and on gene. So how do they do this gene testing? I showed you one example that we can take our blood sample and then measure it, but we have a smarter way to do it now. We just collect. Does anybody know how are we doing this testing? This is just extra bit, guys, okay? Not going to come in exam. Saliva, have you heard that? So we we are asked to give our saliva and then they test genes there. So I got this done, and on your left side, there is a big report. I can show you that also, but just let's stay here because these are the enzymes that we have studied today. So they looked at my MTHFR1, which is that 667 one. Then they looked at that MTHFR2, okay, which is A adenine to guanine 180, A to G. So they looked at both. And then what they found was that MTHFR1 is okay. Green means that homozygous and correct formation of the enzyme. But when it's yellow, it means that there is a SNP. Okay, so instead of A, there is a G. And it is happening only in the one allele, one nucleotide. So what this tells me is that there is 50% efficiency of this enzyme in my body. Okay. Now let's come back to the CBS gene. Remember I mentioned this is the pathway from which I make glutathione. And there is a red here. That means that there is a double SNP equivalent to a mutation. So it is telling me that this enzyme is working at an efficiency of maybe about just 5 to 10%. So already I get this information and then I look at some of the data about dietary interventions. So there are human studies now that tell us that if we are consuming high amount of omega-3, it can modulate gene expression of inflammatory biomarkers. Okay. If we have sufficient amount of vitamin D3, then it can modulate gene expression of interleukins and tumor necrosis factor alpha. We also have human studies that talk about Mediterranean diets, ratio of omega-3 to 6, and high antioxidant diet, and how it can change gene expression of inflammatory molecules. So the data is still insufficient. If you are interested, I can show you the full report as well. So, are you interested? Just don't want to overburden you.
Yes, Nancy, please.
So they are looking at B12. They are looking at blood pressure regulations. And the data is based on whatever human data has been collected. So this is just tip of the iceberg, because you have seen that SNP in any of the enzymes in such a complex body as humans can cause an issue. I always had an issue with my blood pressure medications, sorry, not medication, blood pressure control. And you can see that vascular health, I don't have any green. So what does that mean to me? I need to take more of beetroot to control my blood pressure. I need to work more. I can't do a lot of weightlifting, but I can do more of walking. That will be more beneficial for me, because by doing more weights, I am putting pressure on a system that has few SNPs in that pathway. So they are looking at leptin also. Okay. So people who say that they are not able to reduce weight because they are not able to control their hunger, because there is double mutation here. So So we can then devise strategies. Okay, we know now leptin is not working, so maybe let's work with other genes that are working well and support the pathways there. So many NutriPath are doing it now. Fit Genes is doing it. Bioconcepts have a gene testing. So I encourage you to experiment with this when you come in the student clinic. Maybe, you know, if you have a client or you are becoming a mock client for your colleague, you can at least check MTHFR and have a look.
Nancy, is it expensive to have it done?
Has anybody else got it done? For one, we had that Bioconcept kit for 1 or 2 enzyme. It's about $80. Has any and regarding this Fit Genes one, that costed around 260, I think, for this entire report. And you know, they can use the data from Ancestry also. If you have got that done, then they charge less and they will share the data with Ancestry. So even that is happening.
Nancy, were you surprised by your results? Were you surprised by the amount of SNPs that you had? And
it was something, Kathleen, kind of, I knew it, you know, and but it kind of reassured me, okay, maybe that's what I was doing wrong, that I was trying to control my blood pressure despite having this SNP. So now I know that I have to go an extra mile as compared to my husband, who, in spite of being stressful, his BP is always normal, no medication. So somewhere I knew it, but it kind of gave me a reflection. Okay, that's the reason. So,
what's the NLA under the COX-2 to 4 pathway? What's that there?
where is that one there?
So on what you've got on the screen at the moment,
I don't know what I've got.
omega-3, etc.
Yeah,
Under
yeah, yeah.
2 to 4, what's NLA and A-linolenic acid? Can you see under COX-2 to 4 it says NLA?
Oh, not sure.
What does that mean?
I'll have to go back to the report. No, see, COX-2 and COX-3 we will study in session 10. And
Yes,
you will
yep.
know why.
I know that's the inflammatory pathway. I know that, Nancy.
Yes.
Just wondered
Yes.
what NLA stood for.
Sorry, can't remember.
That's all right, Nancy. No, I knew there was a COX-2 pathway, but I thought I didn't realise there was a I think I had heard of the COX-3, but I hadn't heard of the COX-4. So
Yes. Yes.
no worries. Thanks, Nancy.
That's okay. And so there is now kind of movement toward personalized medicine, individualized medicine, and what we say here is about holistic approach, and there we can look at both nutrigenomics and nutrigenetics. So if we know that somebody has a SNP, then we can work around it, give them a dietary and a lifestyle plan so that we can work around that SNP. Because now we know that some of the foods, they can influence gene expression. We talked about folate a lot today, but has anybody heard about any other food that can change gene expression? Thinking about the food sources of cofactors that we have studied today. Yes, Isabel, choline is right. Choline is one of the methyl donors. Anybody else?
Nancy, I just wondered about like leafy greens because they contain a lot of the B vitamins, don't they?
Yes, carry on. I'm looking for broccoli. Okay.
The cruciferous, is it?
Yes, the cruciferous veggies.
Sulforaphane, is it, Nancy?
Yes, sulforaphane. Yes, they can upregulate NRF pathways. And we are not going in detail, just that extra bit which you can keep a note of. So there's a lot of work around cruciferous veggies, especially liver detox pathways and all. They can work wonders. Omega-3 is another one. What else? Mediterranean diet, high antioxidant, bright color foods and veggies. That's another one. So we'll learn more. We'll continue to learn more. Comments, questions?
I've just got so much on my mind. It's a bit overwhelming, but it's all absolutely awesome. It's great. I was just thinking with that fit gene testing, if you got your partner to do that before you had children, could that indicate, I don't know, I was just thinking could be quite interesting, like with the comparisons to know what could come out in your children. Yeah. Anyway.
Yeah. Yeah, we can.
And Nancy, that, like your report, is that sort of an average amount of SNPs that people might have?
Again, we don't know because we are so different. And I was so surprised that I had an issue with MTHFR. Like, I thought it's okay. And it's different for different people. Like my vitamin D is okay. Glutathione is okay, but blood pressure is different. So again, this is what I said, a very less number of enzymes, how complicated our body is. So some people may have more if they are exposed to, let's say, BPA, or they are working with heavy metals all the time. Bisphenol A is also known to influence gene expression. There's a lot of data. Hydrocarbons in a negative way.
And Nancy, so even though you had that red next to the that pathway that produces the glutathione, your glutathione is okay. Is that what you're saying, Nancy? You know how you had the pathway that produced the glutathione, you had the red, so you had the, you know, it was showing, it was, you said down here, the CBS pathway, you said it'd be working at about 5%, but you've still got your glutathione still okay. Is that what you're saying?
Yes.
So you've got that genetic defect.
Yes, what's this telling me is that this enzyme is functioning at its minimal capabilities. So I can upregulate it by having NAC, N-acetylcysteine, giving a ready-made cysteine in my body, and I can upregulate other antioxidants which are okay in my body. So it is not completely a failure, but now I know that I have to take extra care about supporting this pathway for myself. Does that make sense?
That makes sense, Nancy. So really, like you said, this should be something, you know, when you head off to the doctor, this should be one of the first things that's done, isn't it? And then you can look what lifestyle adjustments you need to make to get yourself well.
Yes, and that's when, you know, last session we were discussing about how yoga, meditation, Tai Chi, all those activities, they can modulate somehow even gene expression. We are still trying to understand how, but when we see benefits in people, we know at some level, it might not be at methylation level. Methylation level is the one where we can make a change through dietary modification, but maybe through other epigenetic mechanisms, they are acting. So certainly we have certain tools, but then as you would have noticed from that post-lecture discussion, that we need to be mindful that it does not go the other way. Okay? Like deciding the fate of somebody just on the base of this information, insurance agencies misusing this data. So just be mindful of those other issues.
We should also be mindful of the language we use around the polymorphisms as well, and not refer to them as mutations.
Yes, Deborah. Yes, I agree.
Because, you know, sometimes they might be a genetic advantage. We don't know everything yet.
See, yes, you know, and that's the reason I thought that we should study more about it, have it in our biochemistry, because there was so much hype around MTHFR only. Maybe because, you know, we have specialized at testing, but there are other things that we cannot ignore. And another point that I wanted to make was that earlier, remember when we were hunter and gatherers, we were storing facts. So it was a positive thing that was happening earlier. But now that same gene is an issue because we have changed our environment so much. So there are actually multiple layers. And yeah. Yes, Jane. Jane is saying that even if we know that SNP, we still treat the sign and symptoms that the patient is experiencing. It can just give us extra insight. Yes, I agree. It can give us some tools. That's what I'm saying. But we have to still look at holistically. We have to look at person's medical history. We have to look at them as individual. What is their occupation like? So yeah, we can just have an extra insight, an extra tool to work around.
Nancy, do you send many of your clients off for this testing?
I know many of my colleagues do, and not yet. I have not done a lot of it till I'm pretty sure that it's something I can't yet manage. I don't. You can get a lot of information from genetic history, family history. So yeah,
Yep.
not yet.
That makes sense, Nancy. Thank you.
Okay, great. Thank you, guys. I'll stop here and hope you all have a good week.
Thank you so much, Nancy.
Yep. Thank you, ma'am.